and welcome to the Amplifying Scientific Innovation video podcast. I'm your host, Dr. Sophia Onoye, founder and CEO of the Sophia Consulting Firm, a Weed Bank certified life science marketing and communications consultancy that was established in New York City with the goal of amplifying scientific innovation. The goal of this podcast is to showcase the importance of science advocacy, health equity, and influential leadership through conversations with senior life science leaders who share a unique leadership journey, corporate vision, and industry outlook. My guest today is Mr. David Barrett, Chief Executive Officer of the American Society of Gene and Cell Therapy, otherwise known as the ASGCT. The mission of the ASGCT is to advance knowledge, awareness, and education leading to discovery and clinical application of gene and cell therapies to alleviate disease. I was connected to David by ASGCT's former Director of Policy and Advocacy, Betsy Foss Campbell, who in turn was introduced to me by BioMarin's Dr. Adora Ndo, a client and fellow Nigerian American woman in STEM. I'm absolutely thrilled to welcome you to the show, David.
Thank you so much, Sophia. It's my pleasure to be here. Thank you for the invitation.
It's my pleasure as well. So I always start the show with the same question, which is, what is your definition of scientific innovation?
Well, thank you very much. It's a great question. And the way that I think about it is two components primarily. The first of which are the significant discoveries, those that are responsible for the leaps and bounds in any field. And in gene and cell therapy, for instance, some of those major scientific advancements and innovations include things like sequencing of the human genome, which has made possible identification of a lot of the genes that can be targeted by traditional gene therapies. Also, the successful human application of gene transfer technologies, and then the discovery of gene editing technologies like CRISPR, and now emerging technologies like prime editing and base editing. So those major leaps forward in any scientific field, I think, make up scientific innovation. But another and perhaps equally important component to scientific innovation is the iterative process, the sheer force of will that many scientists use to move an idea forward over many years, or perhaps even many decades, as we've seen in gene therapy. And I think that type of innovation is impressive and inspiring in so many ways, just that tenacity that so many scientists have in advancing their ideals. And those are the two ways that I really think most about scientific innovation.
Great. I love the balance view, because you understand, as you know, the big sort of giant novel discoveries, but there's also the optimization, the fine-tuning of the existing discoveries. And I think that's what makes your definition really unique. So thank you for sharing that.
Absolutely.
You're welcome. All right. So why did you choose to become the CEO of the ASGCT? I know it's a big question, but I'm curious.
Sure. So I was very fortunate to be selected by our board of directors to lead the society in this position. And I'm really honored to have the opportunity to do so. Why I selected ASGCT as an organization that I was interested in pursuing was a variety of reasons. First of all, at the time that I joined ASGCT, which was in late 2016, I was at a bit of an inflection point in my own career where I was leaving legal practice in a multinational publicly traded organization and in a highly regulated field. And I started to think back on my career and the portions of my career where I felt most useful and most fulfilled, and where I really felt most engaged. And that forced me to recognize, or it revealed to me that the times when I really was most engaged were those times that I was working in the nonprofit sector. And I had been fortunate enough to have experiences earlier in my career working for medical, dental, and scientific societies, as well as the American Medical Association in a health policy analysis role. And I really enjoyed those times in my career. So having the opportunity to make a major change led me to a number of organizations, of which ASGCT quickly emerged as the one that I was most interested in pursuing. One of the other components that I really started to think about at that time is coming from the field that I was in. It was a constricting environment, or an environment where the organization was going through a time in its life cycle where it was getting smaller and more efficient. And I was really interested in being within an organization and in a field that was growing. And that was something that was really important to me, rather than just helping an organization maintain or sustain whatever market position it has, looking at a field that was primed and ready for growth. And so when I came across the American Society of Gene and Cell Therapy, I was immediately interested. And so that was really what drew me into the organization initially. And then as I moved through the interview process, what I was immediately struck by was the engagement of the individual leaders and those that were in the presidential pipeline. At the time, it was Dr. Michelle Kalos, Dr. Helen Heslop, and Dr. Cindy Dunbar, who were in the presidential line. And I found their outlooks on gene therapy and the organization to really be, like I said, engaging. They were interested in advancing the field, but also really interested in advancing areas of the science as it applied to patients. And I found them to be really compassionate leaders in the space. And it, again, drew me into the organization even further and really made me want to be a part of ASGCT.
I love that, Johnny. I believe that no one can tell the story of your life better than you. So you really took us through your unique career journey and how we landed you at the ASGCT at a time where we're at the forefront of the gene and cell therapy revolution. So this is really remarkable. And it's a follow-up to my next question, which is, can you provide us with a top-line overview of ongoing work at the ASGCT, especially as it relates to advocacy and policy, which you briefly mentioned?
Yeah, I can. So ASGCT is active in a number of areas. And some of the areas in which we're most active right now and in growing programs hasn't been on the policy and advocacy front, though that does play into everything that we do. So I'd like to just happily give a top-line approach to everything that we have going on.
Yes.
And the most significant is the component that I think we're best known for, and that's our annual scientific conference. Mm-hmm. The Society has given much thought into how we approach our conference, particularly since the beginning of the pandemic. And we're very excited to be returning to an in-person meeting in May of 2022 in Washington, D.C., with, of course, a hybrid component associated with it. So much of our focus has been in defining what does a hybrid scientific conference mean to our society, our members, and stakeholders in gene and cell therapy, making content available to the widest possible audience, but also creating an engaging environment for those that are able to join us in person. And so that really is where the focus of the majority of our attention is on the staff side of ASGCT. We're also in a unique position with respect to some of our other programs. So one of the other major programs of the Society is the publication of our 4 academic journals, so the molecular therapy, and then its sibling journals, molecular therapy, nucleic acids, MT, methods in clinical development, and then a 4th journal, molecular therapy, oncolytics. And those journals have really increased rapidly and recently in their impact factor and in their impact on the field overall. So the opportunity to publish more good science in the space is something that we've focused on. And then also with the advent of the pandemic, it has forced us to look at how we offer educational opportunities. And I think one of the interesting metrics is that in 2019, prior to the pandemic, we offered, I believe it was 3 co-sponsored webinars over the course of the year. And in 2022, we will offer more than 40 webinars over the course of the year as well as every single bit of live content that we present in person will also be presented digitally. So we've really leaned in heavily to the digital presentation of educational content and scientific sharing. On the policy and advocacy front, one of the areas that we've also leaned into recently is the creation of a quarterly landscape report. And I know many other organizations in the space do something very similar, and we're very appreciative of every angle and every bit of information on the landscape of gene and cell therapy. So what we've tried to focus on is presentation of and some quantification of the preclinical development in gene and cell therapy. Our scientific society is very unique in that we exist at the intersection of science and industry. And that's really the sweet spot for ASGCT. So what we've tried to do in our landscape reports is identify as best we can the preclinical development space and the emerging technologies that are becoming industrialized and where investment is being focused. So that's a new report or a new program in 2021 that we've leaned into. And then to diverge just a bit from the policy and advocacy side of things, one of the areas that I'm most proud of and most interested in is the work that we're doing to fund early career scientists. And again, the juxtaposition from 2018 when we began this program to where we are now is really an enormous amount of investment relative to the size of ASGCT in early career investigators. So in 2018, we launched what we've called the Career Development Awards, which are relatively small dollar amount grants to early career investigators to help them establish independence or advance their own areas within their labs. While it's not limited to just postdocs, it is primarily targeted to postdocs and other maybe early tenure track faculty. And we started with an initial investment of, I believe the number was $280,000 that we had allocated. And in 2021, we will crest $1.1 million in funding for early career investigators. Yeah, I'm really proud of that. Through 9 career development awards, 2 diversity, equity, and inclusion fellowships, and 1 grant into research that into a therapeutic area that disproportionately impacts minorities in the United States. And so through those 3 different award categories, funding a little bit more than $1.1 million in research. Really proud of that.
Wow. I mean, that's truly remarkable. I was a cancer research scientist. I was a postdoc at the Yale Cancer Center. And I can imagine if I got an award like that back in the day, I think that would have been truly phenomenal. And I really appreciate what you're doing by encouraging the scientists of the future, because you have to have those investments for us to really see innovation. And on the publication front, I'm also happy to support your team in sort of advancing the state of science. In this case, is the AAV and gene vectors, but I think there's so much innovation going on in the space. And that's why I really like that focus on publications and such. And finally, you mentioned diversity, and that's obviously an angle that I would jump on any day. So so clinical trial diversity is obviously it's important for optimizing therapeutic efficacy and safety. So can you tell us more about how the ASGCT is leveraging tools such as the clinical trials finders and other programs that you have to improve clinical trial diversity?
Yes, it's so the clinical trial finder, I think, has a direct benefit on, or at least an intended direct benefit on clinical trial diversity. And so that tool is a resource that we've put together that organizes all clinical trials. So it pulls data from clinicaltrials.gov, and the common denominator is gene and cell therapy trials. And to my knowledge, it's the only clinical trial resource where it's organized based on gene and cell therapy. And it goes across all therapeutic areas. And what we've done is coupled that with the creation of that resource with our patient education program, which we're working very closely with dozens of patient advocacy groups to develop resources on gene and cell therapies that are either being developed or soon to be developed for those disease indications. And combining that with the clinical trial finder is an attempt to engage a wider population and more diverse population of potential trial participants.
Wow, that's truly remarkable. I'm very excited about that. And I want everyone, I think as many people as possible, to know about those resources, because that's going to be integral to maximizing the use and also innovating the space. So thank you for sharing that perspective. Now, you've had quite the career. I mean, if I remember correctly, you're a lawyer, you're an attorney by training. So are there any notable mentors that have helped to shape your career journey today?
Yeah, so there are a lot of mentors that have helped shape my career, and many more teachers along the way too. I've had the great fortune to work with a number of individuals who have taught me aspects of how I approach my career, both my professional application of knowledge, as well as how I think about the role that I serve in a job like I have today. So some of those mentors, I recall one of whom was my first boss when I left law school and got my first job as an attorney. And he and I shared a lot of aspects of our career journey. So I did not follow a traditional pathway where I did undergrad and then went straight into law school. I worked for 7 or 8 years before I went back to law school. And I went back while I was working full-time at the American Medical Association. And so I was in a different space mentally, as well as just in my life when I went back to school. And it was certainly a challenge to do so. And it put me in a unique position relative to my cohorts in school. And so finding a mentor who had shared some of those life experiences of he had a career in journalism before he went back to school. He was, married with young children when he was in school, and then not getting out of school, not following the traditional pathway that so many attorneys do, where they, do an internship and then they go work for a law firm as an associate and climb the associate ladder until they make partner. And then at that point, I have a little bit more mobility in their career. I didn't follow that traditional pathway, and he didn't either. And he taught me some very important lessons about how I approach hiring and mentoring people that I encounter in my role as well, and helping to identify talent in a variety of places. And yes, you can identify talent through the traditional means, but there are so many more people who are intelligent and professional and engaged and motivated, and helping to find those people and cultivate relationships at work and in my career with them, and helping to move them up and into spaces where they can flourish really helps to add something to the overall organization. So he taught me some of those lessons that I use really every day.
Wow, I mean, that's really remarkable. Something tells me that part of your journey is also being unique, often off the beaten path, right? You don't do things because everybody else is doing it. And I think that allows you to be the leader that you are at the ASGCT today. So thank you for that.
Yeah, it certainly allowed me to collect experiences over the years, which is something that I think was really important relative to my role. And in collecting those experiences, I think it's given me a viewpoint that's really helped at ASGCT. We serve so many audiences. We have obviously our primary membership, which are scientists and researchers, but we understand that the work that our members do impacts society much more broadly. So we have patient engagement, we have engagement of clinicians and others who are engaged in the delivery of care. And then we also have engagement with the public overall, as well as, as many other stakeholder groups as you can think of. And so having a diversity of experience, I think, helps to think about how our mission impacts those different stakeholder groups.
Great. I love it. Diversity of experience, diversity of thought, advancing the state of science. Now, speaking of science, are there any emerging technologies in the gene and cell therapy space that you're currently excited about, and why?
Oh yeah. Oh, there's so many, right? There's so much going on in this space. It's all very exciting. Some of the areas that I think are most exciting are the areas that are adjunct to the delivery of gene and cell therapy. So the availability of genetic screening and genetic markers of disease, I think is really fascinating, because as we're able to identify patients that could benefit or understand the mechanisms of disease, that really helps aid in the development of personalized therapeutic approaches. And also, particularly as gene therapy has impacted patients, young patients with rare disease, newborn screening and the availability of newborn screening will really help to understand the impact of disease and the potential for these therapeutic approaches, and also be able to identify as many infants as possible that could potentially benefit from these therapeutic approaches. So I think that's really important. And then going all the way to the other side of the spectrum, I'm also really excited about the genetic markers and the biomarkers of the impact of therapeutic approaches, to better understand how gene therapies and gene and cell therapies are impacting patients. And I think that's really important with the gene therapies that are potentially one-time treatments. Understanding how they impact diseases is really important. Now, areas within gene and cell therapy that I also think are really important are regulated control of gene therapies. So this is an area where there's a fair bit of research going on right now. How do we potentially turn on or turn off gene transfer technology or gene editing technology, and possibly even how to titrate the impact of those technologies? That's an area that I think is really important. Other areas within gene editing, I think are really fascinating. There's a lot going on with clinical application, in vivo approaches for CRISPR, and also new approaches to editing like base editing and prime editing. Those are really important. And then I guess the last area of gene therapy that I'll cover is the one that we're really excited about within ASGCT. We have such a diverse membership that our members aren't just working on these traditional gene transfer technologies, but have been working on the use of genetic material to treat or alleviate disease in a variety of ways, and using a variety of different mechanisms. So the use of RNA approaches has been a mainstay within our organization for 25 years, including mRNA vaccine approaches for emerging disease and other disease like HIV. And so obviously we have seen an absolute explosion in the applicability of these types of therapeutic approaches that's having a big impact on our membership in the world. But there's also a growing interest in potentially transient gene therapies, leveraging RNA technology. And that's a place where there's a lot of work going on right now within our membership, and we're very excited about. The last area that I'll touch on is not directly related to gene and cell therapy, but in how gene and cell therapies are advanced and what regulatory pathways exist. And while it's not a technological advancement, I think it's an advancement in the policy sector that has really benefited the proliferation of gene therapies. So the breakthrough designations, the priority reviews, the ways that the FDA is reviewing and looking at and thinking about gene and cell therapies and advancing those is something that I think is very important. And as we watched with the mRNA COVID vaccines, the ability to move therapeutics very quickly, yet efficiently and effectively through the review process is really fascinating to me and all of our members.
Right. I love how comprehensive your approach is, right? You talked about a number of technologies, from enabling technologies to policy changes, right? And I think that showcases just how broad innovation is within this space, and it's rapidly innovating. You can't even keep up. There's just so much going on right now. But I think one of the things that you mentioned that I really like are just those key consideration factors that will be important for sustaining innovation in the life science industry. So there's a more open-mindedness in the side of the regulators and such. Are there any other factors that you can think about that will help to improve or sustain innovation in our industry?
Yes. So in order to improve or sustain innovation, we need to continue to engage the people, the scientists, and have well-trained professionals who are entering STEM professions, and specifically in gene and cell therapy. So I think it's really essential that we continue to feed the pipeline of young professionals and scientists who are entering this field, not just in sheer numbers, but you had mentioned diversity of thought, which I think is really important. The broader and more diverse group of scientists and professionals that we can bring into the field, the more innovative and the more successful I believe that gene and cell therapy will be. Another area where I think it's really necessary to think carefully is in the financial stability in this field in order to continue to sustain innovation. Right? These are expensive approaches, and it requires thoughtfulness and investment in basic science, and then it requires interest among investors and companies and other resources in the translation of those approaches to therapeutic applications. And we've got to ensure that they're that as they are created, that they are accessible to patients. And I think that entire cycle will lead to sustainability in the field. Right. Or at least without it, it wouldn't be sustainable. So we've got to really think carefully about that and remain focused on that financial life cycle. And the last area that I was thinking about with that question is the accessibility of the technology and public education around the technology. And this is something that also the pandemic has really opened my eyes to, the need for public engagement and the need to help not just patients, but anyone who might benefit from gene and cell therapies or from these advances in life sciences to understand exactly what it is. So lifting the veil and explaining how these technologies are created, how they're advanced, and the overall benefit that they have. And by engaging the public and making the technology more accessible, I think that individuals would be more likely to not just enter life sciences as a potential career, but also to think about how these technologies could be applicable in their own lives and be open to new therapeutic options.
Right. No, I love how you sort of showcase accessibility as not just therapeutic access, but also ensuring knowledge access. I think not only would people be more interested in our field, we'll also have more investors to advance that science. So thank you so much.
That's a great point. Yeah, absolutely. Yeah.
Well, my final question is the easiest one. Do you have any other comments or thoughts that you'd like to share with our audience before we wrap today?
Well, it's just such an exciting time in gene and cell therapy. And I don't have anything else that I wanted to add other than just to thank you for the work that you're doing and let you know how much I appreciate learning about this podcast and becoming a listener. It's been really an enjoyable journey for me. And to say thank you again for inviting me. I can't tell you how much I appreciate this. Thank you very much for the opportunity to speak with you this morning.
Well, it's an honor and a pleasure. I appreciate the robustness of your knowledge and the generosity of your time. So I hope that we can reconnect again in the very near future. But for now, I will say bye, but I look forward to connecting again in the future. Thank you.
Thank you so much. Take care.